Hello, everyone. My name is Abdullah Shahada, and today I'm about to present you my research entitled Sequencing the Nucleocapsid of Tomato Spotted Wilt Virus as a Step Towards Understanding the Peanut Breaking Resistance in Alabama. So TSWV is an ampicin single-stranded RNA Orthotospovirus from the family Bunyviridae. So this virus family has both viruses that are able to infect plants and animals. So these share some characteristics between them. The genome organization of this virus, as we can see here in figure 1, is composed of 3 segments. That's why its genome is tripartite. It's composed of 3 segments. The first one is the S segment, stands for small, and then the second one is M segment, stands for medium, and then the third one is the L, which stands for large. The 3 segments, they encode for 6 different proteins for the virus. So the virus was first reported on a peanut in Alabama in 1988. And the virus has a global distribution. It's have the ability to infect over 1,000 plant species. And also, all orthotospoviruses are transmitted by Thrips in a persistent propagative manner, meaning that the virus is able to, once the insect acquired the virus during the larva stage, they are able to transmit it during their entire lifespan, but only as adults. So this is the meaning behind persistence. But propagative means that the virus is able to multiply and replicate within the insects and actually infecting them, but it doesn't show symptoms in them. So as we said, the virus is infecting so many plants and it's causing so much damage. So the peanut breeders, they bred for resistant cultivars, so they can, it's like a management way for the virus. And then those lines been using for a long time, but in the recent year, especially in 2021, 2022, the peanut in the field started to witness a high incidence of the virus. So that's why we did this study, because we wanted to understand why peanut is breaking the resistance and why we are seeing so much symptoms in the field. And also, like when you have we have high incidence, this in the other way mean that you have a huge yield loss. So we wanted to understand that, and then our hypothesis is that maybe the mutation in the genome of TSWV is the reason behind that, and it's pushing the emergence of a new strain that are able to break the resistance of peanut. Therefore, we started by utilizing the nucleocapsid as a first step to understand this phenomenon. And then the reason we chose the nucleocapsid, which is on the S segment, is because the nucleocapsid has, it's the most sequenced gene of this virus. So it has a lot of data available on the NCBI, which means that once we get our sequences back, we can compare it with the references that are on the NCBI, and it will help us to understand and analyze our data. So to do that, we started by collecting 121 samples. And in all the samples that were collected, we collected them from three locations in Alabama, as we can see here on the map. And then also all the samples, they were all symptomatic for the virus, showing atypical symptoms of the virus, which are a yellow ring spot. And those lines here in table 1, those are all the lines that we sampled. And then all the lines that are in red, they are coming from the Peanut RX guide. And this is the institution, the official institution in US that is responsible for breeding peanuts against multiple diseases, including TSWV. So the farmers usually go there and then use this guide so they can select what is the best cultivar that they need for their farm. So all the samples were carried to the lab, and then the whole RNA was extracted. The DNA was made using specific primers, and then the nucleocapsid was amplified using specific primers. After that, all the positive samples were subjected to the purification using the purification kit that we have in the lab. After that, they were cloned in the BJet vector. After that, they were transformed in E. coli component cells, and then we sent 2 samples from each from 2 colonies from each sample for sequencing. And then the reason we did that is because we want to guarantee that at least we get 1 complete N from each sample. So the sequences were received, and then they were analyzed, and then they were converted to protein. After that, the alignment was made to detect the changes. Then, as a final step, we generated and constructed a phylogenetic tree, as we can see here in figure 3. So this phylogenetic tree actually is composed of all Alabama sequences, along with Georgia and North Carolina, and some from Asia. Those are all one that have a complete sequence on the NCBI. So as a result, we found that we have 89 samples were positive out of 121. If we go back to table 1, all the numbers that are on the right for the lines, they refer to how many samples were positive out of how many collected. For example, the first one, there were 3 positive out of 4, and so on. So as we can tell here, these lines that are in red, which are reported on the peanut RX as resistant cultivars, they are not actually resistant. They are very susceptible and highly susceptible for the virus. And the lines that are in black color, they still under testing. They're not released lines yet. Also, the protein alignment for the nucleocapsid, as we can see here in table 2, reveal that there are 7 amino acid conserved mutations among the nucleocapsid sequence. As we can see here, this is the reference genome in the top, coming from the NCBIs. We can see here in a tree, and this is the reference, and this is the genome that represents Alabama. So, for example, in the site 42, we have the amino acid I changed to L, and the change is represented among all the Alabama strain, as we can see here in all of them, except one sample here and one sample here, but the rest of them, all the samples show this change. So we think that all these changes is one reason, not the main reason, but one reason why the virus is breaking the resistance. They may contribute to the virus ability to overcome the peanut resistant cultivars and break their resistance. So the phylogenetic tree also showed us that Alabama strain, most of them, the majority of the Alabama strain are actually very similar and related to Georgia strain. As we can see here, they share the same ancestry roots together. Furthermore, we were able to see 3 subclades in group A and group B and group C. So in group A, there were a blend of sequences from all Alabama strain, along with Georgia and North Carolina, and also all of Asia strains, except this one. So this clade here suggests that T. sabovina Alabama is very variable, and we have so many, like we have big diversity here, more than other than the other locations. And in a group B, we have all the sequences from Alabama. In a group C, yeah. In a group C here, we have BRU, we have GCREC, we have WGREC, only with Georgia, which is support and confirming the hypothesis that most of Alabama strains are very, very similar to Georgia, especially in this clade. The same thing here in group B, but in the absence of the GCREC, it's not found here. But it's only like 2 locations from Alabama, and also again with Georgia. So to furthermore understand these 3 subclades and why they are subclading with each other and what makes them unique to each other, we selected one sample to represent each group. As we can see here, we chose from group A, we chose BRU25, from group C, we chose WGREC5, and then from group C, we chose the GCREC45. So why we chose them? Because we need to localize them in the plant cells using the Nicotiana benthamiana, so we can see if the mutation that they have, which is very unique to them, is going to change the localization in the cells or not. Of course, in a comparison with the reference genome, that based on this paper, it has 2 RNA binding regions. And in the first sample, it has 1 chain that is located in the first region, but the other 2 samples, they have changes that are hanging in the core of the nucleocapsid. So this is the next step that we're going to do, which is actually ongoing now. We're doing it. And then for the first ongoing research, the first one is that, as I mentioned before, we only sequenced the nucleocapsid. Let's go back here. So we only sequenced the nucleocapsid, and we were able to observe 7 amino acid changes. So think about sequencing the whole genome. I think, or I'm guessing that we will get even more. And as I mentioned before, the 7 amino acid changes are not the only reason that contributed to the virus and its ability to overcome the resistant ability of the peanut. So that's what we're doing now. We are going to send all the positive sample, which are 89, for full genome sequencing through the Illumina MySeq instrument at North Carolina State University. So we can also observe and detect the other mutation in the full genome of this virus. As a final point, I would like to mention is that, as I mentioned here before, that the virus may move between the 2 states from Alabama to Georgia. This could happen with 2 possible ways. The first one is that the virus is able to infect so many plants. As I mentioned, its host range is very wide. It's almost over 1,000. So the virus is able to infect any plant that, on its way, meaning that it can be jumping from plant to another or field to another field between the 2 states and spread easily. The second one, the virus has taken a huge advantage by transmitted by Thrips, because Thrips, as we can tell, they have much diversity than any other insect. And also they fly because they have wings. And then also they can be carried by wind or hurricanes. So they can also move the virus, because once they acquire it as larvae, they will transmit it until their entire lifespan. So that's it for me. I hope I was not very fast. And please, if you have any questions, I'm gonna bring the second slide. If you have any question, just email me on those 2 emails, and I will be so happy to answer your questions. Thank you so much for having the time to watch the video, and have a good day.